Welcome to Animal Biotechnology. Today, we are diving into one of the most fascinating and groundbreaking topics in modern science, nuclear transplantation. If you have ever wondered how scientists can create an exact genetic copy of a living animal, you are in the right place. We will build our understanding from the ground up, starting with a simple analogy, moving through the biological mechanisms, and finally looking at the mathematics that scientists use to measure their success in the laboratory. Let us get started. To truly understand how this works, let us start with a simple analogy. Think of a living cell as a highly advanced biological factory. In this factory, the nucleus acts as the master control room, and it contains all the blueprints, which we call DNA. If you want to change what the factory produces, you do not need to tear down and rebuild the entire factory from scratch. Instead, you simply need to swap out the blueprints in the control room. Nuclear transplantation is exactly like this control room swap. We're just giving the existing factory a brand new set of instructions to follow. Now that we have our factory analogy in mind, let us look at the formal scientific definition. In the scientific community, nuclear transplantation is most commonly known as somatic cell nuclear transfer, or SCNT for short. This is a highly delicate laboratory technique. We start with an unfertilized egg cell, which is perfectly equipped with all the cellular machinery needed to build a new organism. The first major step is to physically remove its original nucleus. Then, we replace it with a donor nucleus extracted from a somatic cell. A somatic cell is just any regular body cell, like a skin or udder cell, rather than a reproductive cell. Building on that definition, let us explore exactly how the first step of this mechanism is performed under the microscope. The process of removing the original nucleus is called enucleation. We start with an unfertilized oocyte, which is just a scientific term for an egg cell. Using an incredibly tiny microscopic glass needle, scientists carefully pierce the egg and physically extract its original nucleus. This leaves us with an enucleated egg, a cell that has all the machinery to build life, but currently has no instructions to follow. Now that we have an empty egg cell, we move to the next crucial phase. The enucleated egg is fused with a donor nucleus extracted from a somatic cell. Once this new nucleus is introduced, we have what is called a reconstructed egg. But it will not start growing on its own. The reconstructed egg must be artificially stimulated using electrical shocks or chemical baths. This stimulation tricks the egg into dividing, making it act exactly as if it had been naturally fertilized by a sperm. If successful, this dividing cell mass becomes an embryo that is genetically identical to the somatic cell donor. Understanding this mechanism leads us to ask, why is this so important in the real world? This technique completely revolutionized biology because it proved that adult, fully specialized cells still contain all the necessary genetic information to create an entire organism from scratch. This is the exact method used to create Dolly the sheep, proving mammalian cloning was possible. Today, it is heavily used in the agricultural sector. Farmers and scientists use it to multiply elite livestock that possess superior traits, ensuring herds have exceptionally high milk production or strong built-in disease resistance. Beyond agriculture, this technology has profound implications for other critical fields. In modern medicine, nuclear transplantation is an essential tool for producing transgenic animals. These are animals that have been genetically engineered to secrete valuable human pharmaceuticals directly in their milk, making complex drug production much more efficient and scalable. Furthermore, this technique holds massive potential for global wildlife conservation efforts. For highly endangered species teetering on the brink of extinction, scientists can use preserved tissue samples to clone new individuals, potentially saving entire species from disappearing forever and restoring genetic diversity. While the biological applications are incredible, scientists must rigorously track their success in the laboratory. Because this process is highly prone to biological failure, we use a mathematical metric called cloning efficiency. This formula calculates the ratio of successful births to the total number of laboratory attempts. E represents the cloning efficiency as a percentage. N live stands for the total number of live, healthy offspring born. N recon represents the total number of reconstructed embryos initially created. If the number of live births increases while the attempts stay the same, the efficiency goes up, indicating a successful protocol. Conversely, if you have many attempts but very few births, the efficiency drops significantly. Before we move on, let us see this formula in action with a concrete example. Imagine a biotechnology laboratory attempting to clone a transgenic cow. They successfully create 250 reconstructed embryos, which is our N recon. After the gestation period, they record a total of 5 live births, which is our N live. We want to find the cloning efficiency. We substitute 5 and 250 into our equation. Dividing 5 by 250 gives us a decimal value of 0.02. Multiplying by 100 converts this to a percentage. The final answer is 2%. This physically means that for every 100 embryos painstakingly reconstructed, only 2 survive to become live calves. Now that we have used this formula to find efficiency, let us look at how we can manipulate it for planning future experiments. Sometimes we already know our target efficiency and how many live animals we need, but we do not know how many embryos to start with. We need to algebraically rearrange our formula to solve for N recon. First, we start with our standard efficiency equation. Next, we multiply both sides by N recon to get it out of the denominator. Finally, we divide both sides by E. This isolates N recon on the left side, giving us a new, derived formula that allows us to predict the exact number of laboratory attempts required to guarantee a specific outcome. With our newly derived formula in hand, let us apply it to a conservation scenario. A team wants to clone an endangered wild sheep. They know the cloning efficiency for this species is 1.5%. They absolutely need to produce at least 3 live cloned sheep to establish a breeding herd. We plug our known values into the rearranged equation, 3 for N live and 1.5 for E. Dividing 3 by 1.5 gives us exactly 2. We then multiply by 100 to account for the percentage scaling. The final answer is 200 reconstructed embryos. This means the team must perform nuclear transplantation 200 times to statistically guarantee their goal of 3 live sheep. To solidify everything we have discussed and calculated, let us look at a visual schematic of the entire process. On the far left of the diagram, you can see the unfertilized egg cell undergoing enucleation. Notice the microscopic needle physically removing the original nucleus, which is marked with an X. Moving to the right, we see the nuclear transfer step, where the new somatic nucleus is carefully inserted into the empty egg. Next, the lightning bolt symbol represents the electrical activation, which is the artificial stimulation required to kickstart cell division. Finally, on the far right, the process results in a dividing cluster of cells, which is our successfully cloned embryo ready for implantation. We have covered a lot of ground today, so let us summarize the most important ideas. First, nuclear transplantation, or SCNT, is a profound tool that allows us to completely bypass natural sexual reproduction to create exact genetic copies. Second, we achieve this by replacing the nucleus of an egg cell with a somatic nucleus, effectively reprogramming the egg to develop into a clone. Third, as we saw through our calculations, the process is mathematically inefficient, requiring hundreds of attempts for a single success. However, despite this inefficiency, its applications in medicine, agriculture, and conservation make it an absolutely essential technique in modern biology. You now have a solid understanding of both the biological mechanics and the statistical realities of nuclear transplantation.